This video will focus on the genetic code, how genes work, and how it is that genes can code for specific sequences of amino acids in proteins. After watching the video and thinking critically about the concepts that are presented, you should have a basic understanding of the genetic code. You should also be able to explain the so-called central dogma of molecular biology, and you should be able to identify various types of mutations. So first of all, the concept of a gene. At the most basic level, you can think of a gene simply as a sequence of bases in DNA that codes for a particular protein. It's a little more complex than that. There might be other sequences, regulatory sequences, and so forth. But in a nutshell, just think about a gene as the DNA bases that code for proteins. If you think about how this works, what we see here in the sketch is a eukaryotic cell. So here's a nucleus within a much larger cell. Think about the DNA within your cell, so chromosomes, for example. They, of course, contain specific sequences of bases, and when a gene is expressed, what happens? Well, one of those strands of DNA is sort of used to create a complementary piece of RNA. That's shown here as a yellow ribbon. So a piece of RNA is produced, it then exits the nucleus. Now it's out here in the cytosol. It then interacts with ribosomes. Ribosomes sort of move their way along that RNA, and they use that RNA to produce a particular protein, shown as a green ribbon here. Of course, that protein has a specific sequence of amino acids that's determined by the sequence of bases in this RNA. That, of course, is determined by the sequence of bases in the DNA. So in terms of how this works, you can think about the RNA as a so-called messenger of information, carrying that genetic information in the DNA out here where it's used to produce a protein. So in this case, we could talk about this RNA as messenger RNA or mRNA. There are other types of RNAs as well that play different roles within cells. In this scenario, what we're talking about here is this first stage where DNA is used to produce a complementary piece of RNA. That's the process of transcription. Then the sequence of RNA bases is essentially translated by ribosomes into a protein. So that's the process of translation. So you probably have heard of transcription and translation before. That's what those processes refer to. And then collectively, this whole idea of this flow of information from DNA, shown here in red, to RNA, shown here in yellow, to a protein, shown in green. That's the so-called central dogma of molecular biology. And again, the sequence of bases in DNA codes for a complementary sequence bases in the messenger RNA, which is then used to link specific amino acids together when a protein is produced. So just a few things I'll comment on while this figure is up. First of all, notice the directionality. Your 3', 5', 5', 3'. That's important. We have to keep track of the 5s and 3s from here on out. It really doesn't matter which way you draw a piece of DNA, but you always have to keep the directionality in mind. So in this case, if the 3' is toward the left here in the DNA, the messenger RNA that's produced, it will be anti-parallel. So this end would be the 5' of the RNA. Right? So again, you have to keep that in mind. Also, if you take a look at this figure, recall that RNA incorporates the base uracil, indicated by U here, instead of thymine. So typically you might be tempted to think, oh, the complement of A would be T, but recall RNA incorporates U instead of T. So you have to keep that in mind. In addition, if you notice the RNA, we can identify chunks of free bases. So CGC, for example, AAC, CUG. So those sequences, or those chunks, those clusters of free bases, those are referred to as codons. And keep in mind that every codon codes for a specific amino acid. So CGC codes for amino acid arginine. AAC codes for the amino acid asparagine, and so forth. So let's think about how this works. What we have here is a sketch illustrating a piece of DNA in red. So recall DNA double-stranded, anti-parallel, like we see here. When a gene is expressed, just one of those strands is one that's used to produce a protein. So let's just say, for example, that it happens to be the bottom strand. It doesn't really matter. Again, in this case, if it is the bottom strand, we see, well, that's the template strand. That would be transcribed with sequences that are complementary. So TA, AU, CG, CG, and so forth. And again, keep that 3', 5' in mind. That's important. And then notice if these sequences are compartmentalized into these sequences of 3 bases, these codons. It just so happens in this example, this first codon, AUG, happens to be a so-called start codon. That's profoundly important. We'll come back to that in a second. But that's important because once you find your start codon, that then sets the reading frame. In other words, from there on out, every sequence of 3 bases represents a codon. So in this case, GCC is one codon. AAU, that's another codon. And you sort of keep going and keep producing amino acids until you encounter one of these so-called stop codons. UAA happens to be one of these stop codons. So let's explore that in a little more detail. So here is a table illustrating the genetic code. So how this works, you notice we have sequence of messenger RNA bases here. So these are all codons. So UUU is a codon, UUC is a codon, and so forth. And notice, for example, these 4 codons, they all code for the same amino acid, leucine. These 4 codons, they all code for the same amino acid, proline, and so forth. So that's the genetic code. But let's explore this a little bit farther. You have to keep, remember I keep saying you have to keep in mind your 5s and 3s. Codons are typically written in a 5-3 direction, referring to the messenger RNA that that is in question. So for example, the codon, or no, it doesn't matter, CAU, that would be 5' CAU 3', right? So again, that's, you have to keep your directionality in mind. How does this work? Let's look at an example. So I give you a sequence of DNA bases up there. Recall DNA would be double-stranded. So I'm just keeping it simple and giving you the single strand that's used to create a protein. So if you see a sequence like this, you might be asked, well, what is the sequence of amino acids that would be produced in a protein if that gene is expressed? Now, what you have to do first is find one of those start codons. So if you look at your genetic table, well, there's the start codon, AUG, and it codes for methionine. So that's step 1. Actually, I take that back. That's step 2 after you know your RNA that you're dealing with. So if you see a DNA sequence, your first step would be to essentially transcribe that into a complementary piece of messenger RNA. So in that case, it would look like this. Again, keep your 5s and 3s in mind. So the complement of the DNA would be CGG, UUAA, and so forth, right? That's your RNA sequence and the messenger RNA. Then you have to find your start codon in that messenger RNA. So you can't just assume that these first 3 bases, that's going to be the beginning of your protein. What you have to do is find out where that start code, oh, there it is right there, AUG. So now you've identified the start codon. So that sets the reading frame. In other words, once you identify that start codon, every 3 bases after that represents a separate codon. And you keep reading those codons until you encounter a stop codon. So notice UAA, that's a stop codon. There are a few others as well, UAG and UGA, right? Those are all stop codons. Now you've identified the so-called coding region of this gene. Then it's a simple matter of seeing which amino acids would be produced. So start codon, AUG, that gives us methionine, so there's met. Then AAA, that gives us lysine, so LYS. GUU, that gives us valine, so there's VAL. AUU, that gives us isoleucine, and then our stop codon. So this sequence would be transcribed and translated into this sequence of amino acids. So that's how the genetic code works. Notice if you look at the genetic code, we can talk about it as being redundant and specific. redundant in the sense that for many of these amino acids, there are multiple codons that can code for the same amino acid, as we see in leucine and proline and several others as well. And that's kind of important because the implications is that you can have mutation, for example, that could change the last base in either of these codons, and it doesn't really change the amino acid that's produced in the protein, right? So that's important as well, but it's also very specific. In other words, every codon codes for a very specific amino acid. So that's an important outcome as well. So if you think about DNA replication, we've got lots of proteins and enzymes involved in that process, and they typically work pretty well and pretty faithfully make effective, exact copies of the DNA bases within our chromosomes, right? But mistakes can occur, and we can get mutations in a variety of different ways. So let's just think about mutations for a few moments here. So what happens when genes are disrupted? Again, that can occur in various ways, but let's just first think about point mutations. So we're talking about mutations at just a single base pair, or could be a few base pairs, but sort of small scale mutations. What are some of the potential outcomes of these types of mutations? Well, this figure does a nice job at summarizing a variety of point mutations. So just as reference to begin with, let's just say we have a sequence of bases in DNA, shown here in red. That's our our beginning point. That's our original gene. When that's transcribed, we get this sequence of bases in messenger RNA. We see the codons there. And when those are translated, we get this following sequence of amino acids shown in green. So that's just our beginning reference point. The first mutation we'll focus on is a silent mutation. So let's just say we're going to mutate this gene here and change this T to a C. Well, ultimately, yes, that's going to change this codon, but it turns out that both UAU and UEC, they both code for tyrosine. So we say that's a silent mutation because there's no change in the amino acid sequence of that protein. So perhaps no real implications in terms of the protein that's produced. So that's one possibility. It's also possible that we might have mutation that does indeed change an amino acid. So a so-called missense mutation. So we're going to change this G to T. Ultimately, that's going to code for cysteine instead of tryptophan. So it's changed the amino acid there. Is that going to have a huge impact on the protein? Well, it just depends. It might, but it might not. It just totally depends on the situation. Another possible mutation is a so-called nonsense. This could be a bit more dramatic. So we're going to change this G to an A. What that does is converts UGG into UGA as a stop codon. So that's a big deal because now our protein is a lot shorter, right, than what it was to begin with. So this nonsense mutation could have a huge impact on the ability of the protein to function properly. The other one we'll mention here is a so-called frame shift mutation. Frame shift mutations essentially happen when you either insert an extra base, like we see here, and now our whole sequence is one base longer, or you can also have a base be removed. In that case, the whole sequence would be one base shorter. The implications could be pretty dramatic. So because what that does, that sort of offsets your reading frame. So if we insert a base, if we change this TGG, and now we're going to insert an extra base, we're going to insert a C. Now, from here on out, everything after that has a different reading frame than we had to begin with. So ultimately, you're probably going to see a big change in the amino acids that are linked together compared to the situation in the original version of that gene. So again, the implications could be quite significant. So that just gives you a sense of some of the different types of point mutations that can be found in living organisms. And that concludes our brief discussion of how genes work.